Hello and good day, whatever time it is for you. This is the Module 8, Genetics and Genomics. Here are the learning outcomes for Genetics and Genomics. We will be going through this, and I've decided to split these into sections. So for this one, we're going to have 4 sections. First is going to be why should you care and what is a genome? The second is how a genome changes. The third is going to talk about classic genetics. And the fourth is going to talk about the molecular biology revolution, and that's probably going to be the largest section. I think it's going to make it more easy to digest all this stuff by doing it in sections. So you'll see these videos posted in their sections in Canvas. First of all, why should you care? Bacterial genetics and genomics has had a profound impact on your life, even if you didn't know it. The first genomic revolution came with the discovery of the central dogma and the realization that we could clone useful genes from any source and make as much DNA and protein as we wanted from them. This resulted in the ability to engineer plasmids and digest them, but it was still a little bit of a clunky and slow process, and it took a lot of work to try and get any gene cloned and expressed to do anything with it. The second genomic revolution came with the advent of PCR and DNA sequencing, and this is 1980 to the present. Once we were able to easily sequence DNA, mapping genes became trivial, and PCR made it possible to amplify any sequence that we wanted as long as we knew what the flanking regions were. And then we could get huge amounts of this. It made cloning and getting genes and making alterations quite a bit easier, but still a very intensive process, and that it was a low frequency event, so you had to find the desired mutations that you wanted. The third genomic revolution is taking place right now, and that is the CRISPR revolution. The big thing that CRISPR does is it takes that low frequency event and makes it a 50%, a 50-50 occurrence. This then allows genetic manipulation at a level we've never experienced before. The other thing is it makes these kind of alterations possible in any organism. So this is pretty amazing stuff. These technologies affect your life. One of the major ways it does is in genetically modified organisms. And one of the main instances of this is Roundup Ready crops. These crops have taken over American agriculture. They actually have some benefits because if you use a Roundup Ready crop, it is resistant to a pesticide that will kill most other plants. Because that, you don't have to apply as much pesticide, and you can grow more food in an area. There are some downsides to Roundup Ready crops that you can find out if you read on the internet, and I think those are legitimate concerns, some of them. There are biologicals and drug productions. Many types of drugs are now possible because we can clone genes and easily express almost any protein that we're interested in. An early example of this was serotonin. My brother had carcinoid syndrome when he was a young man, and one of the things that prolonged his life quite a bit was the production of serotonin, and this was one of the first genetically engineered proteins that was made. There are all sorts of enzymes that you use in your daily life that are produced via biotechnology and genomics and genetics, and we're starting to see payoffs in the treatment of disease. Some examples of the companies that use these technologies are shown here. There are pharmaceutical companies such as Upjohn, and there are agricultural companies such as Monsanto. There are food companies such as Danone and General Mills. And then there will be fermentation companies such as Miller Coors that use these technologies. So hopefully in this you've seen that this is really important. It impacts all sorts of facets of your life. You can even go to 23andMe and get your genome analyzed, and it will tell you all sorts of things about yourself. I think in the future, that's going to be a common thing that you actually do at your doctor's office. So genomics and genetics impact you in numerous ways. Now, if we're going to talk about genetics, the first thing that we have to do is figure out what a genome is. So I want you to sit down and think about, for just a second, what you think a genome is. Now that you have your answer, here's what I think a genome is. It's the complete genetic content of a cell or organism. For bacteria and archaea, what this means is their chromosomes and their plasmids. For eukarya, it is their chromosomes, if they have plasmids, most of them don't, and then the organelles chromosomes. Bacteria and archaeal genomes are usually between 1.5 and 14 megabase pairs. That's 1.5 to 14 million base pairs. The largest is Serangium cellulosum. So that's a pretty large range. Eukaryotic genomes range from 2 megabase pairs to 23,000 megabase pairs. An example is Saccharomyces cerevisiae that has 14.27 megabase pairs. The human genome is 3,200 megabase pairs. Microbial genome structure varies. Some will have only a single chromosome. Others will have chromosomes or several chromosomes and then a number of different plasmids. So what's the difference? Chromosomes have essential genes like RNA polymerase, the ribosome genes. You can't survive without this piece of DNA. Copy numbers vary between organisms, right? You can have haploid, which means you have one copy of it. You can have diploid and polyploid, which is obviously seen more in eukaryotic genomes. Plasmids carry non-essential genes. You can lose the plasmid and the organism can still survive and grow. Copy numbers vary from 1 to hundreds of copies, and you can have different plasmids per cell. So one cell can have more than one plasmid. For bacteria, the chromosome is usually circular and usually about 1 to 12 megabase pairs. But of course, there's always exceptions, and here's some examples. Mycobacterium tuberculosis. By the way, you do not need to remember this slide. These are just examples. What I want you to get from this slide is that genomes can vary. Some will have a single plasmid, as in Mycobacterium tuberculosis. Others will have all sorts of different things. Agrobacterium tumefaciens. Agrobacterium tumefaciens has a circular chromosome, a linear chromosome, and then 2 circular plasmids. Borrelia burgdorferi has a linear chromosome and 21 plasmids. So it can be very different just depending on the organism. Another thing I want you to understand about genomes is that bacterial and archaeal genomes contain mostly coding sequence. So coding sequence is at a premium. Eukaryotic genomes are full of non-coding sequences. And you can see this in this figure, is that all the green areas here are coding sequences in E. coli. Right? And in a human genome, if you look at the same 50 you look at another 50 kb region, you can see there's lots of non-coding regions and then these little slices of coding regions. So it's very different. This brings us to plasmids. Many microbes also contain plasmids, and this is more of a bacterial and archaeal thing. It is unusual for eukaryotes to have plasmids, and yeast are a notable exception. These are circular, double-stranded pieces of DNA. There are examples of linear plasmids, but most of the ones we know about are circular. Plasmids can direct their own replication, but they will use the host machinery to actually do the copying. So they direct it, but then use the enzymes of the host to do the replication. They range in size from 2 kb to about 1,000 kb, and they encode accessory functions that are not essential for survival under most conditions. Again, that's a very important distinction between a chromosome and a plasmid. Shown here is a plasmid that's commercially available from Promega, and you can see a number of important features. First of all, there is the origin of replication. This is where the replication machinery binds and starts copying of the plasmid. There is a gene here called ampicillin. This is an ampicillin resistance gene. It's actually called beta-lactamase. And then there is a multiple cloning site, and this is a site where it has a bunch of restriction enzymes where you can clone things into it. So some examples of the accessory functions that plasmids encode are shown here. And you can see that there's a wide range of different things that can be done. One of the most important is conjugation. So conjugation functions are found in a wide range of bacteria, and this will be important later on when I explain what conjugation is. There are plasmids that are used in antibiotic production. There are metabolic functions. There are resistance to antibiotics and toxic chemicals. And then there's a whole class of virulence plasmids. I don't expect you to memorize this chart. I just want you to know that drug resistance is not the only thing that plasmids encode. They can actually encode a whole variety of different capabilities for the organism. As I talked about for the replication of plasmids, they'll use the host replication machinery. One example of how this is done is here's the origin of replication. A rep protein will bind. This will cause the replication machinery to come in, DNA polymerase, single-stranded binding proteins, helicase, etc. All that comes in, and then it makes a copy of the plasmid. The origin of replication is essential to this process. These origins are somewhat specific for host species. So if you have a specific original replication, you only find this plasmid in specific hosts. Other plasmids have a broader host range and a more broad origin replication that can be recognized by a bunch of different microorganisms. Finally, the one thing I really want to emphasize is plasmids are selfish DNA. You can consider them like a virus without a capsid, right? They infect cells, they grow in the cells, and a lot of the time the bacteria doesn't want them there. Having a function that the microorganism wants to use makes them more valuable, but they really are parasites on their host. Another thing that the plasmid has to do, besides replicate itself, is make sure that it's carried on through the cell division cycle into the daughter cells. And this is called plasmid segregation. In this, the goal of the plasmid is to make sure that each daughter cell gets one. For low copy number plasmids, what they'll often do is they'll coordinate with chromosome replication. They'll use the same cytoskeletal replication apparatus. It gets divided between the cells and goes through. For high copy number plasmids, it's really not a problem because random partitioning occurs. If you've got 40 copies of a plasmid inside a cell, some of them are going to go into each side. One interesting example is the toxin-antitoxin system. So you'll find this in a number of different plasmids. You'll have a promoter. It synthesizes a toxin and an antitoxin. So the toxin and the antitoxin. These will come together, and the antitoxin blocks the activity of the toxin. The antitoxin is actually not as stable as the toxin, and over time it gets degraded by LON or CLIP proteases, and then the toxin will attack its cellular target. So if this, this is getting synthesized at a low level all the time, you always have plenty of antitoxin around to block the toxin. However, if you lose the plasmid, now you just have the protein sitting in the cytoplasm and there's not new antitoxin being made. This antitoxin degrades, and now the toxin is active and hits its cellular target. Therefore, if you lose the plasmid, it's toxic to the cell. So example targets that the toxins will attack include DNA gyrase. Some toxins will degrade mRNA, some toxins will inactivate translation, and some toxins will go after peptidoglycan synthesis. So this is kind of a clever system that plasmids use to prevent their loss. We finish this section with a clicker question. You sequence a new bacterium's entire genome and assemble it into 4 circular pieces of 3 megabase pairs, 1 megabase pair, 0.7 megabase pairs, and 100 kilobase pairs. Which ones are chromosomes and which ones are plasmids?